Hello. Um, this is back again to my channels, to my lectures channels, science channels, as I intend to introduce these lectures from my channel to help students and to educate people as well who doesn't know about all these things that we learn in school. I hope to explain this in both local language and in English. And today our topic is going to be the diversity of life and their basic similarities. We look at life as we can see things, like we can see us ourselves as humans, we can see the animals, we can see the plants, the trees, but there are things that we cannot see, which are also part of life. And these are the things that we need to discuss today. And we need to explain how similar they are and what are their basic differences and similarities. In doing so, we're going to have a look at their cell structures and we're going to have a look at their DNA. Now, to start, we can see that the breakdown of life is too vast. Life is too vast. There is a lot of living things around the world, in the oceans, in the air, on land, under the ground. Life is everywhere. So to understand it, people who study life, they are called taxonomists. They break it down into kingdoms. And these are the five basic groups that I have listed here. You have bacteria, you have protists, fungi, plants, and animals. And animals include us. And underneath there, I can say that despite their diversity and their variation, all life share the basic signature, which is DNA. DNA is the oxyribonucleic acids. This is what all life is made out of. Nothing can be living without having DNA in it. Rocks are not living, they don't have DNA. The sand, the soil, they don't have DNA. Water does not have DNA. These things do not live. The things that live are only the things that have DNA in them. And this is what we're going to explore today. Now, I start with, as I said, I have listed bacteria and protists, which are part of life, but are living things that we cannot see. So for our examples, I have chosen bacteria, and the bacteria I have chosen is Bacillus cereus. Because maybe back home we are more familiar with rice using cooking rice. Bacillus cereus is a bacteria that is found in rice, and if rice is not cooked properly or is not stored properly, it can develop this bacteria. And bacteria develops and divides in every 10 to 20 minutes. If you start with one bacteria, in every 10 or 20 minutes, that bacteria will divide and become two. And the next 10 to 20 minutes, that two will become four. And the next 10 to 20 minutes, that four will become eight, and so on. So 8 goes to 16, 16 goes to 32, 32 goes to 64, 64 goes to 128, 128 goes to 256, and this is how it goes up and multiplies in the food that you have. And it becomes so infectious and can cause illness and food poisoning. So that is why I chose Bacillus cereus. There are different types of bacteria, but Bacillus cereus is the one that is prone to be found in rice. Now, I explained about protists as well. Protists are different forms of life as well. It's categorized as a different form of life. It's a unicellular organism. It is a one cell, single cell organism. And the protist I chose today is the one that we can associate with our livings back home, which is Plasmodium. Plasmodium is a protist that creates malaria, and we all know what malaria is. So this is what creates malaria. There are different types of protists, which we will explain a little bit later after this slide, which causes malaria. There are different species of malaria. Malaria is not only one. There are different kinds of malaria, and their severity depends on also which kind of malaria you have acquired. So it's an acquired illness. It's not contagious. You cannot pass malaria from one person to another. It doesn't matter whether you are in contact with that person or not, you share the same food with that person or not. Malaria is not passed, it's not contagious. It is something that you acquire from a mosquito bitten. A mosquito is Anopheles mosquito, which is the female Anopheles mosquito that feeds in blood. The male mosquito does not feed in blood. It feeds in honey and nectar and in flowers. So it has something to do with the transmission of malaria. Okay. So here I have listed the types of malaria that you can have. The first one is the um, Plasmodium falciparum. This species is responsible for the majority of malaria deaths. It's a very devastating malaria, very severe one. You have Plasmodium vivax. This species is the most widespread and can cause severe infections, including fatal ones as well. It can also kill. You have Plasmodium ovale. This species is characterized by eosinophilic dots or syphilinous dots in the cytoplasm of infected red blood cells. It is in the red blood cells. So all these are different types of malaria that can happen, that you can get infected with, and they all have different type of treatment. It's not that all malaria can be treated with one specific thing. There are some that can be treated in combination with other things. Um, since this is not a topic to discuss about malaria, it is just an example that I was trying to bring up. So we just got to move on. Right. I discussed about also fungi. That also is also another is an other type of living thing, which is what you are seeing here now. Fungi, there are different species of fungi as well. This is mushrooms, which have also a diversity of species of mushrooms that can exist, but they are all mushrooms, but they are different species of mushrooms. Like you can have different fish, different species of fish, different species of dogs, cats. You can have different species. All living things have different species. And this is the different kind of species that are listed here for mushrooms, but they are classified as fungi. Okay, you have yeast as well. Yeast is used in the making of bread, and it makes the bread to inflate and rise up. So it's also another living thing. You can see the yeast here as a powder. You can just start to wonder, is is any life in this dry powder or something like, but believe you me, yes, there is life. When it is dry like this, it hibernates, so it incubates itself, and once it started to get water and get wet, it wakes up straight away and become live and starts to do whatever it has to do. So that is yeast, is also another family of fungi. Now, all of them, the similarities in all these living things that I showed you there. I just showed you the protists and the fungi and the fungi, because we don't see them most of the time, and the bacteria, they are microscopic, but we can see ourselves as humans. We can see animals. We can see plants. This is why I haven't given you examples of those, because we know them. But what is similar in all of them is the DNA. DNA is the picture that you're seeing here. This is how it is. It is in a spiral form with with the with the sides of this of the spiral being phosphates of sugars, and you have the rungs here. That is the ladder. Like they are adenine, thymine, guanine, and cytosine. This is how they are linked together. They cannot link together in any other way other than this way. Always adenine has to link together with thymine, and guanine has to link together with cytosine. Me, the way I remember is ATCG, which is at CG, at GC. Once you can remember that, or at CG, then you can remember the connections between the base pairs. These are called the base pairs, and it is what the sequence of every living thing is. How they are arranged is what make difference between living things. All living things have the same base pairs, but the way the base pairs are sequenced, they are arranged, is what makes them different. This is why you have the sheep becomes a sheep, the cow become a cow, the human become a human, the fish become a fish. It's because of the way the base pairs are sequenced. And it is the base pairs that makes the codes, that makes the proteins, that makes you, me, sheep, cow, fish, and whatever you can think of, any life form that you can think of. It is the code, the DNA, that makes the amino acids, that makes the codes, that codes for the proteins, that makes the cells, that makes the tissues, that makes the organs of that living thing. So DNA is the main thing here. And you can see it here again, being labeled as adenine, thymine, cytosine, guanine. And the phosphate backbone is what it is what spirals around this. This is the phosphate backbones. And that is what DNA is. Right. But before we get any further, I would need to give a background explanation of DNA. And in doing so, we cannot go without discussing the father of genetics. This is Gregor Mendel. He is the father of genetics. He is the one who introduced all our modern genetics today. And how did he do that? Mendel was a monk. He was born in a German-speaking family in the Silesian part of the Austrian Empire, today known as Czechoslovakia, and he gained recognition as the founder of the modern sciences of genetics. He went to study at the University of Vienna in Austria, but he did poorly in his university studies. He couldn't make it through. His lecturer labeled him as not really fit. He lacks insight. So Mendel failed in his university studies, but then he went back to his home where his father was a farmer, and then he started to work in the farms to work on genetics. He was influenced by Carl Unger, who was the biologist at that time. This was, you're talking about 1865. Mendel was born in 1822 and died in 1884. But in around 1865, that was when he started his plant hybridization to try to understand how heredity works. So all this discussion today is about heredity. How do we inherit from our parents? This is what Mendel was thinking about. How do we inherit from our parents? Is it a fluid that transfers from the parent to the offsprings, or is it a piece of pebble, or what is it? He doesn't know. And what he wanted to know was to do an experiment that could at least tell him how people inherit, or how generations inherit from their ancestors. Now, to do this experiment, he cannot do it on people, because if you want to see how a person will inherit and pass the inheritance to the next generation, it will take years for you to complete that experiment. For example, if you have a child, you observe what the child has inherited from the parents. but that child has to take at least 9 months before the parent give a birth to that child. That new generation comes in, and then you have to observe the characteristics that the person has inherited from the parents, and then you have to wait for that child to grow up to maturity where they can give birth to another child, which is the other generation. So you can observe and see again that generation what they have inherited from their parents. So that would take a long time. That experiment may not even find you alive. You might be dead by then. But then Mendel knows that there is a unification theory between botany and zoology. What happens in botany? Botany is the study of plants. So what happened in botany is exactly what happens in zoology. Zoology is the study of animals. So these are two branches that have been divided to study living things on this planet. Botanists will study the living things on plants, how they evolve, what is their DNA, their characteristics, and how plant species occur. And zoologists will start to study about animals, about humans, about other animals, how DNA affects them, how they evolve, how they transpire. So there is a unification between these two because of they all have DNA, and the DNA in plants and the DNA in humans are all the same. The nucleotides, these are the small molecules that are linked together in the ladder, is all the same. They are all adenine, thymine, guanine, and cytosine. Wherever you go, whether you go to a bacteria, or whether you go to a plant, whether you go to a protist, whether you go to a fungi, whether you go to an animal, these are the nucleotides that you find in their DNA. As I said, the way these nucleotides are arranged is what makes them different. That is the sequence. That is called sequencing. That is why now um, they are trying to sequence all living things. And the Human Genome Project that was concluded in 2003 was, was to sequence the human genome to know exactly how many nucleotides do we have in the human genes. And the result came out to be 300 million, I don't know, 3 billion genes. 3 billion nucleotides are in there. And but out of the 3 billion nucleotides, you have 20 to 30,000 genes that coded for the proteins that makes you and me. And there are other genes there that are called regulatory genes. Those genes, they don't code for proteins, but they regulate the genes that code for proteins. When the gene that is required to code for a given protein, for example, for a gene that is to make your nose, if that gene is to be activated to start making your nose, then the regulatory gene will switch on, and that gene will know that it's time for it to start working on making your nose. That is called gene expression. That gene will start to express, and the regulatory gene will keep it expressing, keep it expressing, keep it expressing until it reach the maximum where your nose should be. Whether you are a nose of a human, a nose of a cow, a nose of a crocodile, it depends on what animal is producing that nose. And then when it reaches to that level where it's required to stop, the, the regulatory gene will switch off, and then it stops producing. So this is how proteins are created. And this is the job of the regulatory genes that helps the genes that make the proteins that make all of us. Okay, so now Mendel, what he wanted to do was to see how heredity works. So his experiment, because he can't do it on humans, then he thought he can do it on plants. And the plant he chose was garden pea. Okay, so now we can see this is a garden pea. We all use green peas in our food that we use today, like mixed vegetables and things like where you have mixed corn, mixed sweet corn, and mixed peas, mixed carrots and things like that. The peas, the green peas that you see there, they are the garden peas. Then that is what Mendel chose to do his experiment. He got seven characters to look at, how those characters are passed from one generation to another. He was able to look at the form of the pea, the seed itself. With the seed, he can have two things that he can look at. In the flower, the color of the flower, he can only look at one type of thing, whether the flower is white or whether it's violet. The other thing is the pot itself, where the peas are enveloped, what form the pot is. These are the type of things that he was looking at, which one. And then the stem as well, he looked at which garden pea, short stem versus talling stem. There are short stem peas and tall stem peas. So he got seven characters under which he can use his experiment to see what can transfer from one generation to another. So at the end, he ended up choosing talling stem versus short stem. So he hybridized the tall stem and the short stem peas, and then he grew that pea. When he grew that pea, the pea grew up, and there comes the first generation. You see, the first generation comes quicker because it's a plant. It cannot be as long as a human, bearing a child over 9 months and then having a birth. But this is just a pea, a garden pea. It grows quickly, and then he can observe it and to see what happened. So because he chose talling stem versus short stem, and he hybridized those two, he was trying to see whether um, the offspring that is coming is going to be an average between the two, high and tall, the high and the short. Which one is it going to be, or is it going to be an average? But to his surprise, the first generation came, and all the stems were tall. And then he's like, where is the short? Because I hybridized the short and the tall. How comes the first offspring, the first generation becomes all tall? He couldn't understand. Okay, so he take the first generation and fertilize the first generation by itself, because plants can fertilize themselves as they are hermaphrodites. They can fertilize themselves. Plants, they cannot walk around to go and look for a partner that like animals and humans can do. So nature has endowed them with becoming hermaphrodite, so they can fertilize themselves. And when we come to talk about pollination, we will discuss all about that. But for now, let us just know that plants can fertilize themselves. So he fertilized the first generation by itself, and then he grew it. So lo and behold, the next generation that came, he found that every three there is one, every four there is one short one and three tall plants. So the short started to be seen in the second generation and not in the first generation. So you can see, then Mendel knew that inheritance can skip a generation. And it doesn't mean that you inherit from your parents. Sometimes you can inherit from your grandparents. And this I have seen many times. An example that I can boldly give is my brother is married to his wife. They have a child who has blue eyes. My brother doesn't have blue eyes. His wife doesn't have blue eyes. So where did the blue eyes came from? But I can remember that his wife lived in the same street as I lived back home in the Gambia. And I have seen the family of his wife, one of their sisters had blue eyes. So it seems that the blue eye came from um, the wife's side, the generations of the wife's side. That is why their son had a blue eye. So if you don't know these things, you can maybe even accuse your wife of going to bed with someone else, which is not you. And that is not the case. It is just the lack of understanding of how inheritance work and how it can skip generations. So then Mendel published his book widely to France, to throughout the world. He published his book, and even the biologist that he was relying on, who was teaching him about cells, when he published his book and discussed his findings with that biologist who was called Carl Unger, he was dumbfounded. He didn't even know what Mendel was talking about, because at that time nobody knew what Mendel was talking about. Right. It was something new to science. But still, Mendel knew that the inheritance can skip generations, but still, he did not know the pathway. How exactly does inheritance pass from one generation to the other? He doesn't know it. He just knew that it can skip a generation, but he doesn't know how that can happen. As he published his books, it went worldwide. At the end, Mendel was appointed the abbot of a monastery, and when he died, they burned down, the new abbot that came burned down all his books, because at that time the monastery was very much a rival to science. They don't want to hear what science is saying because it was against or contrary to what they were preaching, so they burned down all his books. But then it was just too late, that because Mendel has published his works worldwide, and that is the experiment on plant hybridization that was done in 1865 and 1866, and that was the result that Mendel got from the experiment. Okay, now so it went now from 1866 till 1931. Nobody was able to be able to examine and look inside the cell very closely to be able to see um, what is happening inside the cells, where the chromosomes are, and inside the chromosomes, where the DNA is. We did not have the magnification required to give us that information at that time. And this is, you see, the period from 1865 to 1931. There was nothing, no instrumentation that could have been found that could give us this information. So here now, I could say that nature has endowed us with a limitation. Our seeing is limited. There is a limit to what we can see, and there is a limit to what we can observe. Our hearing as well, there is a limit to what we can hear, and these are limitations that we can always counter by using instrumentation, by using tools. Science, this is what it does. It uses instrumentations and tools to extend our senses so that we can observe nature more in depth to understand exactly what is going on. Right. As I said, the electromagnetic spectrum under which the radio waves are going, under which television waves are going, under which gamma rays, under which X rays are going. That is the electromagnetic spectrum. We can only see a little portion of the electromagnetic spectrum. And with a part of that, we cannot see infrared, we cannot see ultraviolet, we cannot see all those regions of the electromagnetic spectrum. So therefore, that is why instrumentation is required. But one thing we need to understand in science is that before an instrumentation is developed, it takes a long time. And then science is waiting, it cannot do anything, it cannot observe anything, so it cannot say anything. Because science works with facts. If it doesn't have the facts, it cannot pronounce anything. And to have the facts, you have to have the instrumentation required to give you those facts. So, but to develop the instrumentation takes a long time. And when the instrumentation is developed, then scientists can use it now to make some observations of nature. And also, it takes time for the observations to be made and to be concluded and to be agreed upon and to verify it and to make sure that what they observe is the right thing. So the combination of these two things makes science to take a long time to come up with an answer or to come up with an evidence of something that they are proclaiming. So that will conclude us to say that then every generation has a limit to what they can know. Um, and to a limit to what they can see. And it is the next generation that who can take it from them. What our past parents didn't know, our grandparents, grandparents did not know, today we know. They did not know. Today also there are things that we know that our future generation, there are things that we don't know that our future generations may know. So we cannot deny knowledge to the future generations. We can never say that nobody will be able to see this or nobody will be able to do this. That is a fatal mistake. We always have to make room to let future generations do their part by using the information that were picked up by the older generation, like what we have known today, is there. And if we cannot complete it, the next generation will pick from us, and with further instrumentation, with new developments of observation tools, they can continue to see or to develop things that we could not have seen. And this is what happened with this observation of the cell. It was in 1931 that Ernest Rosca and Max Knoll developed this electron microscope. The electron microscope is a microscope which uses a beam of electrons to magnify an image object and resolve features that are millions of times smaller than what can be seen with the light microscope. The light microscope is the microscope that we use with a light that we can view things. That microscope also had a limit to what it can see. It can magnify up to 2,000 times the size of the object, and that is where it can stop. That's the limit. But then the electron microscope can go further. It can go to 2,000 times the size of the object that you are viewing. So therefore, it was now time for this microscope, the electron microscope, to enable us to view and see inside the cell, to see the chromosome, and even to see up to the DNA level. And that is when these two gentlemen, James Watson and Francis Crick, in 1953, at the Cambridge University, at the MRC department, the Medical Research Department at the Cambridge University in 1953, discovered the structure of DNA. And this is their picture here. And this is James Watson on the right, and this is Francis Crick on the left. Okay, so this is what they discovered. This is the human gene. That is the DNA in the human being. You can see the backbone, which is the phosphate and sugar backbone, and you can see the ladder here, which are the rungs, which are connected by the molecules, which are the DNA molecules, which is adenine and thymine, cytosine, and guanine. These are how they are connected. And when this DNA unwinds in a copying process, it unwinds and becomes two halves. And each half will already attach the other missing half to it, and then they become two. So you have two copies of the same thing. So DNA is a DII, is do it itself. It's not a DIY. Do it yourself. It's a DII, which is do it itself. It is doing it itself. Nobody is telling it what to do. It does it itself. And it can be observed doing it. Now, the DNA of a human is the same as the DNA of a plant. This is, still trying to show the similarities with, with different life forms, as they all contain DNA. So as this is a plant DNA, you can see that also it has the spiral nature of it, which is the phosphate and sugar backbones, and you can see the base pairs of it as well, as arranged as the human one, which is adenine, thymine, guanine, and cytosine arranged together. And you can see the sugar and phosphate backbone here, exactly as you can see it in the humans and animals. And this is also for bacteria and for viruses and for protists. They all have this arrangement. Although viruses may not have DNA, some of them have DNA, some of them have only RNA, which is ribosomes. ribonucleic acids. They are not deoxyribonucleic acids, they are ribonucleic acids. So the only difference within them is the oxygen that is deoxidized from there, and it is on the other one. Deoxyribonucleic acid, which is DNA, doesn't have oxygen, and the ribonucleic acids have oxygen in it. And the arrangement there is also different. In DNA, you have adenine, thymine, guanine, and cytosine, but in RNA, you don't have thymine. It's replaced by uracil. So you have adenine, uracil, guanine, and cytosine. Those are the only two differences that you can find between RNA and DNA. And the RNA is mostly in viruses. It's also in humans, because when we go to explain about the transcription of DNA, maybe that is where RNA should be explained. Right. This is also a bacterial DNA. Just to show the similarities again, you can see that the rungs are there. And then these are the adenine, thymine. You can see it, cytosine, guanine. You can see it faintly written there. And it's the same arrangement that that bacteria have as well. Not different, not anything different from that of the humans. Okay, you have it here as well for viruses. It's the same thing. And then you come to the cell structure. The cell structure, also, I urge you to ignore all these things, the nomenclatures around. What I want you to look at here is the nucleus, which is the center of the cell. The nucleus is where the DNA is located inside the chromosomes, and this is the human cell. That is it has a nucleus, and you can see that it has also a mitochondrion. The mitochondrion is something that we can discuss later, because that is where energy is produced, the energy that makes you walk, the energy that helps you to make movement, the energy that makes your heart beat, the energy that makes your liver works, the energy that makes your kidneys filter all the toxins in your body, the energy that as I have explained in my last lecture on continental drift, I discussed about energy, that nothing can be done without energy. So the body needs energy to do all the work that it's doing and to enable you to physically work as well. So that energy will be discussed in another lesson where we have to discuss more about the mitochondria. But for now, I'm concentrating on the nucleus where the DNA is. So that is the human one. Here you have a plant one as well. It's a plant cell structure, and you can see that the center of the plant also you have the nucleus, and that is where the DNA is. Similar also to that of the animals one. Here you have a yeast one, which is which is a protist, and also there's a nucleus there where the DNA is. Okay, here you have the bacteria and a virus cell structure. They all have this DNA inside here, inside them. But this one is a prokaryote. There are prokaryotic life and eukaryotic life. A prokaryotic life does not have mitochondria. The eukaryotic life is the only life form that has mitochondria. We'll discuss that in the evolution of life when we come to that topic. But for now, I just wanted to show that the DNA is concentrated in the center of it as well. But this one is not doesn't have a nucleus, doesn't have a cell nucleus. It just have a nucleoid in the center of the DNA. Okay, now to conclude, I just want to show you the genomic timeline. Since Gregor Mendel in 1865, this is all that has been happening in the investigation of the DNA up to 2003, where the Human Genome Project was completed. I will leave you with this, and you can zoom into it and you can study it more to understand the advances that were going on during the exploration of DNA and what we can do with it. That is the end of the lecture. The other thing that I wanted to say was that now the Human Genome Project is doubling up efforts to um, to have the genomic sequence of all life forms on this planet, all life forms. And there are many contributing bodies that are helping out in this to explain, to contribute to the genomes of all life. And it is the affiliated project network. It's called Earth Biogenomic Project, the Earth Biogenomic Project. That genomic project is where everybody can contribute as to the genomes they have found, because the genomes are different in different regions of the world. The animals are in different regions of the world. So there are participants in different areas of the world who are participating in trying to sequence the animals that live around there, and to contribute the sequences to the project. So many task forces are working together to find this out. There are 50 different projects that are going on. If you go, I'll leave a website down for you to access to see um, the the Earth Biogenomic Project, what genomes they are after, and to list some of them. There are insect genomes, bird genomes. They're even doing the African biodiversity. For us, it is a shame that we cannot do our own. They are coming to do ours. The African flora and fauna, they are trying to find out all the genes in all the animals, all the plants in African flora and fauna. Amphibians, they're checking, um, aquatic symbiotic animals, eukaryotic marine biodiversity animals, bees, bats, butterflies, crabs, crustaceans, including lobsters, including langoustines, and all these things. They are trying to find out. They are working so hard to um, to put together all the genes of all the animals in the world. So it is going to be very easy, once that is completed, for you to refer to something and exactly know what is what. And I think they're leaving us behind. And it is a shame that we are being left behind. We don't understand what is going on. And it is high time that we have to involve in understanding the nature around us. Look at, for example, the protist that I was telling you, that is the Plasmodium protist that came from the malarial infection. um, where are they? How can we find out where they are? Our environmental department or our Ministry of Health should have been able to categorize the areas which are largely infected with mosquitoes. What kind of mosquitoes are there? There are different types of mosquitoes, but which ones are there that produces or that creates this malaria? What type of malarias are there? This information should be known by the public, by the general public. There should be environmental factors. The environmental agency should go around having a look at waters, testing the waters around the pools, the whatever water is around where mosquitoes can nest, to study it and to understand what is happening there and to inform the general public. That is only one thing. Now, look at this. They are doing our African biodiversity for us. We don't know what is going on. That is why we are just sitting back and just saying, well, let them do this. They cannot do this. Let them do that. Let's see how they can do it. Without us contributing nothing, we are just here criticizing and making objections without trying to do anything to contribute. That is why we are not respected anywhere in the world. And, well, I can't go any further because otherwise it would lead me to politics, which I don't want to go into. This is a science program, and for that matter, I think I end it here. Thank you very much for observing.